All right, welcome back. We are on our first of our processes in chapter 10. This is going to be mitosis. And the last slide will take a minute to talk about binary fission, which is how bacteria or prokaryotic cells divide. So it's way different because they don't have a nucleus. It's a lot easier. All right, so we're going to start off just jumping right in with the first steps of getting ready for mitosis. So I'm going to pick up with G2 of interphase. So if you remember the cell cycle, we have G1, the cell's just doing its thing. S, the DNA is dividing. G2, we are interphase. So the cell is growing, everything else is dividing, but we're still in the visual, it just still looks like interphase. We can't really distinguish between any of the phases of interphase. But what you want to point out when you're looking at these things like in the microscope or a picture of a microscope, since we don't get lab this time around, is you can usually see a clearly defined nucleus, like I see here. This pink cell appears in animal cell example. The brown ones are plant cells. And typically you can also find a clearly defined nucleolus. The nucleus is well defined. You don't really see any obvious chromosomes. Everything is just kind of loosely, again, that chromatin, just kind of loosely organized, but is at this state that the DNA has been replicated. We just can't see that. The centrioles have been replicated, so that happens also during G2 of interphase. We have a nuclear envelope and the chromatin is just loosely coiled. All right, so that's G2. The signal gets sent. We are getting ready to go into M phase, and so let's take a look at that first phase. I'm going to combine prophase and prometaphase. Sometimes some resources call them early and late prophase. Your book calls prophase and prometaphase. I'm just going to combine it all as like one big process. So there's a lot of things that are happening in prophase. It's probably one of the busiest of all of the phases of mitosis. So there's lots of things that happen. So one, we start seeing the nuclear envelope start to break down. So we don't see a clearly defined nuclear envelope, right? So it might be breaking down over here. The next thing we see are the chromosomes are starting to develop. They're starting to supercoil, right? So they're not in that loosely formed chromatin anymore. They're starting to supercondense. And so we can really start seeing those as these dark lines. See, I'm using white and I'm saying dark, but I want it to show up. So we can actually see the chromosomes separately from, it's not just a solid kind of shaded area. We're starting to see some physical things showing up. We also start seeing the centromeres. Let me get on my pen here. We're starting to see these guys, the centromeres, divide or move to opposite ends of the pole, and the spindle fiber is starting to develop. So lots of things are happening. So nuclear envelope is breaking down. The centrioles are starting to migrate. The spindle fiber is starting to develop. The nucleolus is disappearing, right? We don't see an obvious nucleolus anymore. And the chromosomes are just kind of, they're starting to show up. So there's a list of the things that happen in prophase. Okay? And by the time we get to the end or late prophase or prometaphase, we can see there's no nuclear envelope at all. The chromosomes are stuck to their spindle fibers. They're still not lined up or organized or anything, but the spindle's fully formed and the chromosomes are attached. So we have this structure here called the kinetochore. The kinetochore is just like an attachment point, that little light blue color there on the replicated chromosomes. That spindle fibers, the microtubules of the spindle attach to that kinetochore. It's like an anchor. That's where those spindle fibers are hooking onto both sides of that replicated chromosome. In the process of metaphase, metaphase is where the replicated chromosomes get kind of wiggled around and they line up on the middle of the cell, and this is called the metaphase plate, sometimes a plane. The mid-plane. So our centrioles are opposite ends. The spindles are attached to the chromosomes at the kinetochore. That happened in prophase. And now they're all lined up. And this is probably one of my favorite phases to find under the microscope because you can really see a nice line in the middle of the cell. So here is, I'll get a yellow. So here's an animal cell. and here's the chromosomes lined up in the middle. Here's a plant cell, and here's the chromosomes lined up in the middle. Middle, M, metaphase, it works out. So that's a metaphase. It happens very quickly. It does not take a lot of time for metaphase. They just line up and then they move directly into the very next stage, which is anaphase. So anaphase is the microtubule part of the spindle that's connected to the kinetochore. It starts shortening. And some other parts are, so we have shortening this way, and then these guys right here in the middle, these guys, they overlap and they are actually pushing themselves further apart. So they kind of build and push away. So the chromosomes are getting pulled away to opposite ends of the cell. This is another really fun one to look at in the microscope, because again, you can actually see the chromosomes being pulled apart, right, in the animal cell and then here in the plant cell. They're physically being pulled apart to opposite ends of the cell, and that's anaphase. A for apart. That's how I always remembered it. And in the last phase of mitosis, we have telophase or telophase. And this is the replicated chromosomes have been pulled apart. Remember, they're sister chromatids when they're held together. And then when they're pulled apart, they're called chromosomes again, individual chromosomes. We can see the nuclear envelope is starting to form. Nuclear envelope is starting to form, kind of opposite of prophase. The chromosomes are starting to uncoil, so we don't really see the individual chromosomes anymore, kind of like what we see over here in the plant. The spindle is breaking down, and then we start seeing the true division of the cell, which is going to lead into the last step here, which is not technically part of mitosis, which is called cytokinesis. So in telophase, the nucleus is reforming, the nucleolus will show back up, the chromosomes are uncoiling. What else? I think that's it. So that is our telophase, and that's physically the end of mitosis. Last step is called cytokinesis, and this is the physical separation of the cell. Mitosis is the division of the nucleus, which we were successful at the end of telophase. In cytokinesis, depending on what type of cell you are, you're going to have a different process happening. If you are animal cells, you're going to do something called the cleavage furrow. It's basically the cell is splitting in half. It's physically pinching in half, and that will be your two new cells. If you're a plant cell, you have a rigid cell wall. You can't pinch in half. So what you actually start producing is a cell plate, which will eventually become the brand new cell wall in between the two daughter cells. And here we can start seeing that cell plate starting to form between the newly divided nuclei of that plant cell. So in an animal cell, cytokinesis is visualized by cleavage furrow. In plant cells, cytokinesis is visualized by the forming of this new cell plate, which will eventually become the cell wall. Okay, so here's all together now of the processes of mitosis. So again, interphase is just, it could be in any one of the G1S or G2 phases. We can't really tell from a picture. And then when we're ready to go, G2, we get the signal to jump in. Prophase, prometaphase are breaking down of the nuclear envelope, the breaking down of the nucleolus, the condensing of our chromosomes, the spindle is starting to form, and the centrioles start moving to opposite ends of the cell, and the spindle starts attaching to the kinetochores of the replicated chromosomes. In metaphase, all of those replicated chromosomes are lined up in the middle of the cell on the metaphase plate. In anaphase, they're being pulled apart from each other. So now they're replicated chromosomes. The chromatids are being pulled apart into individual chromosomes. And then lastly, we have telophase, which is kind of the reverse of prophase. Nuclear envelope forms, nucleolus reappears, the chromosomes condense, and the spindle fiber breaks down. And then lastly, it's not pictured here, but cytokinesis would be the physical separation of the rest of the cell components. And then you're done. You successfully went from one cell to two identical copies. The DNA just makes identical copies in that S phase, and you have two clones of the original cell. Right? That's mitosis. That's not too bad. And then the last slide here is just showing you binary fission, which takes place in bacterial cells. So in your bacteria, they typically just have one circular chromosome, not linear chromosomes. Like ours are just a single, like a line of DNA. Theirs is actually a continuous circle. So they have on their chromosome what's called the origin of replication. Here it's highlighted as ORI on that little picture. So that's where a kind of what's called a replication bubble forms. So like a little opening happens. and the DNA replication just starts going away from that origin of replication all the way around until it meets up back again. So it'll go in this direction and it will go in this direction until eventually you just get two identical copies of the chromosome. They pull apart, you get this cell wall, very much like the cell plate in a plant cell does, and then voila, you have two cells. Now, in eukaryotic cells, mitosis might take, anywhere from 18 to 30 hours, plus or minus, depending on what kind of cell you are. So it takes a long time for a eukaryotic cell to go through this process of mitosis. Bacteria, because they only have one chromosome, they don't have to deal with a spindle or the nucleus or anything like that. Some of the fastest replicating bacteria like E. coli or Staphylococcus aureus can replicate in 20 to 30 minutes. So that's a generation time in bacteria. So you double your population every 30 minutes. That is a crazy amount of bacteria. Most bacteria average around 1 to 3 hours for their replication and binary fission. So that's just some comparative numbers there for you. It takes a lot longer for eukaryotes because we have a lot more stuff we have to deal with. Bacteria, they're just copying their one chromosome, split, and then they're done. All right, that's our mitosis. I will see you next time for some concepts in prepping for meiosis, and then we'll go through the process of meiosis. Okay, bye.